Okay, welcome back to the Classics of Immunology Journal Club. And before we get started, I just wanted to remind you to like the video. Also, click on the bell, get notified when there's another video to watch, and scroll down and check out my website. In particular, click on the Writing tab, and that'll give you all the classic papers of immunology from starting in 1798 with Edward Jenner. Today, we're going to focus down on in 1980. We're progressing along, but a little bit more slowly than we were before. We were used to do one every few years or every decade. Now we're there's more and more papers now. So in 1980, we published a paper that came out in Nature, in October in Nature, and the authors were myself and Kevin Gilbride and Margaret Favada. The title is Lymphocyte Activating Factor Promotes T cell growth factor production by cloned murine lymphoma cells. And just to give you a little background, of course, you know me, Kevin Gilbride was a new postdoc in the lab, and Margaret Favada was a technician, a relatively new technician in the lab, but she was really gifted in cell culture. She picked up on everything right away. She was fabulous. The questions that we approached at this time, we knew both lymphocyte activating factor and T cell growth factor would activate lymphocytes. The standard assay for lymphocyte activating factor, as you may recall from previous journal clubs, in particular, Egal Gary from 1972 had two papers in the JEM, really defined what people started then calling lymphocyte activating factor. He thought that it was a macrophage product, although he never really ever proved that, essentially. But it was sort of an accepted thing that it was a macrophage product. Now, the problem arose when we put this in our T cell growth factor assay, which you may recall was made with the long-term TCGF dependent cell lines, CTLL1 and 2. LAF didn't do anything. All the cells died. So we knew that whatever LAF was, it wasn't T cell growth factor, that's by definition. So then the question became, well, maybe, but LAF was clearly mitogenic for thymocytes. And so the question is, why is that? And in the introduction, we lay that out, and we say that, the cellular origin of both LAF and TCGF still hasn't been proven beyond a shadow of a doubt. We thought that the TCGF came from T cells, which is why we called it T cell growth factor. Although T cell growth factor says its name, it caused the T cells to grow, but we didn't know it was coming from T cells. We just assumed that it was coming from T cells based on a lot of our experiments. So this paper was focused down on trying to answer that question. And the question was, does LAF possibly promote T cell growth factor production from T cells, by T cells? And that was the question. So in the materials and methods, they're pretty straightforward in this paper. The first thing we needed to do in order to really get at the question of the cellular origin of T cell growth factor, we thought that the best way to do that would be to see if we could find a T cell line, a continuously growing T cell line that would make T cell growth factor. And so we screened through multiple T cell lines, both human and mouse, to see whether or not we could identify, and these were all leukemic cell lines, leukemic lymphoma cell lines. And so we screened through with and without adding a mitogenic lectin to the cell cultures, and then taking the supernatants and looking in our TCGF assay to see whether or not any of these things would make TCGF. And we came upon a cell line that was called WEHI-7. WEHI stands for the Walter and Eliza Hall Institute down in Melbourne, Australia. And WEHI-7 was originated from a BALB-C mouse that had been irradiated. So it was a radiation-induced T cell lymphoma from the BALB-C. And we found that if we just took supernatants from growing cultures of WEHI7. They weren't making any TCGF, but if we added either CON-A or PHA, then they would make readily detectable TCGF in our assay. So that was good. And then we needed a source of lymphocyte activating factor, LAF. And so, we just followed the recipe that was in the literatures, and we used human peripheral blood mononuclear cells, which, is a mixture of lymphocytes and monocytes, activated them with lipopolysaccharide, LPS, as the sort of the standard stimulant to make LAF, and cultured them overnight, and then took the supernatant, filtered the supernatant through a micron filter, and then dialyzed it against normal saline, and then stored it at 4 degrees. So that was it. So that was the source of our lymphocyte activating factor. Then the TCGF assay, as you recall, we reviewed that a little while ago with regard to the long-term continuous cytotoxic T cell lines. So in the results of this paper, after we'd screened WEHI7 for TCGF production, now I'm going to move into a screen sharing situation. Okay, so this is figure 1 from our paper from Nature in October of 1980. And what this plot is a Probit analysis of T cell growth factor assay results. And you can see on the y-axis there we have the CTLL tritiated thymidine incorporation plotted as a percent of maximum. So this goes from 10% up here to 90%. And you can see what the Probit paper does is that it sort of favors the data points around the 50% point, and then has a larger distance between the ones that are at the extreme ends of because it's a bell-shaped curve essentially. And so you get very nice straight line plots as I pointed out when we reviewed the TCGF assay paper. And so this is the results of stimulating WEHI7 cells with concanavalin A without and with the addition of LAF, the LPS stimulated conditioned medium from PBMCs. And you can see that there's a shift to the right in these curves with the addition of LAF. And this is basically about a 4-fold, if you look at the 50% point, about a 4-fold increase, 4 to 5-fold increase in the amount of T cell growth factor activity. And so the point of this plot is to show that the T cell growth factor production by this lymphoma cell line was LAF concentration dependent. So, and we already knew that the growth of the CTLL was TCGF concentration dependent. So that was a very important finding in this paper. Now, figure 2 shows us the LAF concentration dependent production of T cell growth factor from a productive. We cloned the cell line, and this is just representative. About 50% of the clones would make TCGF, and 50% of them would not. This is just a selected clone 49 in the closed circles, and the open circles was a non-productive clone 8, and this was a reciprocal of the LAF dilution from a very diluted 1 to 500 down to 1 to 1 essentially. And you can see that the TCGF activity is very much dependent upon the amount of LAF that was placed into the culture. That's it in terms of the data. The discussion in this paper it was the following. So it looked as though, since the production of TCGF was LAF concentration dependent, and the amount of LAF that you would get out was also concentration dependent on the stimulus there, that what we were really dealing with in this instance was what I called a bimodal amplification system that was ramping up the amount of T cell growth or T cell proliferation that you got out of these cultures, which meant to me that if you could find pharmacological agents that would suppress either LAF or TCGF production or the activity of these molecules, you would have a very potent immunosuppressant compound. And we had already shown in a couple of papers that are on the website from 1979 that glucocorticoids, which were a well-known immunosuppressant used in the clinic for autoinflammatory, autoimmune diseases, allergy, as well as treatment for leukemia, lymphocytic leukemia and lymphoma, that glucocorticoids would block the production of TCGF. So we did a whole series of other experiments that were published in another paper in 1980, which is on the website as well. That was published in the Journal of Experimental Medicine. Basically, there what we show is that glucocorticoids inhibited the production of LAF. So, and we already knew that it inhibited the production of TCGF. And there was a series of experiments in this paper that basically nailed down the coffin on this whole thing as to number one, that LAF is a macrophage product, and LAF can markedly augment TCGF production and subsequently then T cell proliferation. So Joe Oppenheim, who was at the NIH, and he was a LAF guru, he had pointed out in previous publications that the kinds of stimuli, for example, were adjuvants. Lipopolysaccharide was the sine qua non, but that also the polyribonucleotides, poly-IC and poly-AU, and I had experimented with them, and I already knew that poly-IC and poly-AU would activate macrophages to activate T cell proliferation before I got into T cell growth factor. So it all made sense to me. And I felt that probably that's just one of the major ways that immunological adjuvants that everybody used experimentally, as well as in the clinic, to potentiate T cell immune responses. We're working through this LAF TCGF bimodal amplification system. And therefore, in the discussion of this paper, we also proposed that you could use LAF and TCGF to study both the production as well as the activity of both of these cytokines. And because we had cloned lymphoma cells and so forth to study this, we felt that we had our hands on a very productive experimental system to begin to be able to dissect and expose exactly how the T cell immune response was generated. So there was a meeting in Switzerland that occurred in May of 1979, so before these papers were published. But at that meeting, I gave a presentation and showed the data from all of these experiments that we had done. And that was basically a revelation. The title of this meeting was the Second International Lymphokine Conference. So everybody who was anybody in the business was there. And my presentation and so forth created quite a stir, because it really showed with definitive data for the first time that LAF was a macrophage product that acted on T cells to make TCGF, and then the TCGF would cause the cells to proliferate. And so what happened at this meeting was that we'd meet all day long and also after dinner in the evening, but then late at night, we would all repair to the bar and discuss things and so forth, because there were all these scientists who were not allowed to talk all day long, except one of them presenting to the crowd. So they all wanted to talk and so forth. And so we talked late into the night, into 2 and 3 in the morning, about whether or not what the meaning of this whole bimodal amplification system was. And it was proposed, I don't even remember who proposed it, but it was proposed that we make up a new name for these lymphokines and number them, just like in the complement system. There's complement 1, 2, and 3, and so forth, because we sort of assumed that the molecules that were responsible for this activity, all these activities, were probably very similar chemically, biochemically. But we didn't have any data on the biochemical characteristics, or not a lot of data on either LAF or TCGF, people were madly trying to purify these things from conditioned medium. And it wasn't easy because they were basically present in the conditioned medium in very low protein concentrations. Be that as it may, we made up a new nomenclature for these lymphokines, cytokines. That we said, well, they seem to be sending messages between white blood cells of different varieties, like the macrophages and the lymphocytes and so forth. So we decided to call this new class of biochemical activities, which is what they were at this stage of the game. They weren't molecules yet, but we're going to call these biological activities interleukins. Inter meaning between, and leukin is Greek for white blood cell. Interleukins, between white blood cells, essentially. And so because of the fact that it looked as though LAF was coming from macrophages and working on T cells to make T cell growth factor. And if you didn't have any T cell growth factor, you could have LAF, but it didn't have any T cell growth factor activity. So it looked as though LAF was working before TCGF, causing the T cells to make TCGF. And then TCGF was the mitogenic agent that was causing the T cells to proliferate. So we proposed, and ultimately after the meeting wrote a letter to the Journal of Immunology, telling people that there was a bunch of investigators at this meeting that all agreed, some kicking and screaming and others not, on this new nomenclature, that LAF was going to be called interleukin 1 and TCGF was going to be called interleukin 2. And that's the derivation of this whole thing. And so the upshot of this, everybody thought this was wonderful, and this nomenclature was grabbed onto almost immediately by everybody that we knew and so forth. And one upshot of that whole thing was that subsequently, when other people would publish papers that had anything to do with interleukin 1 or interleukin 2, our letter to the editor of the Journal of Immunology was published in the JI, in the Journal of Immunology. And so when they wanted to refer to IL-1 or IL-2, they just referred to that letter. And it was just simply a statement that this is what we're going to do here. There was no experimental data in this letter. Rather, the experimental data were our papers from 1980, the Nature paper and the JAM paper, which they didn't refer to anymore. So TCGF sort of faded away into the distance and nobody referred to it anymore. And so they didn't refer to us either. So that was sort of a bummer. But what can you do? As the beat rolls on and the field was now really taking off and moving fast. And so there wasn't anything I could do about it. And so I said, okay, whatever, we're dealing with interleukin 2. And just a funny story about that is that several years later, after we had purified interleukin 2 and showed everybody what the molecular characteristics were and so forth, I told my kids, who were teenagers at that point, young teenagers, sort of junior high school kids. And I said, well, I discovered the first interleukin molecule. And they looked at me and they said, yeah, sure, Dad, that's why it's number 2. And so I'll leave that with you. Don't forget to like this video and click on the bell and check out all the other papers that are on there, the JAM papers there and this paper, as well as the glucocorticoid papers. And next week, I think we're going to be reviewing a paper from Martin Rollinghoff and Hermann Wagner about the effect of cyclosporin A on LAF, interleukin 1, and TCGF, interleukin 2 production. Thanks. It's been fun.